much for the introduction, Ulrich, and thanks very much for inviting me here. It's a great pleasure to be here. So yeah, the title here really is, why is this all so complicated? We've already heard a lot earlier today about all the issues surrounding the treatment of snakebite with antivenoms. What I want to do is to approach this topic from the angle of snake biology. What is it that causes this great variation in snake venom composition that makes the global snakebite problem such an extraordinary challenge to address? Oh, I did click something. Yeah. But perhaps we need to start thinking about that from the perspective of, advance the slide. Yep. From the perspective of the people who are affected by this. So here's a farmer in West Africa plowing his field, no shoes, as you may see. And as part of that work, this farmer has a high probability that at some point or another, he or his son in this case, might get bitten by one of the snakes that lives in these areas, which is Echis ocellatus, the West African saw-scaled viper. Now, assuming, for instance, his son gets bitten, you're now in a situation where this farmer knows that something that's likely to be life-threatening has just happened to his son, and he now has to confront a considerable number of challenges to address this life-threatening situation. Oops, that is not what I wanted to do. Like this. Here we go. He has to find transport to hospital. We've already discussed a bit the time taken to get to hospital in many remote parts of the world. There may be a cost involved in that, and this is going to be affecting some of the poorest people in the world. He needs to be able to afford the cost of hospital admission. Okay. He needs to address the problem of the cost of hospital admission. Some of the poorest people in the world living on $1 a day or less have to try and afford hospital. Then there's the big question, can the hospital do very much? Is there any antivenom? Can he afford the antivenom if there is it? And after that comes perhaps the cruelest aspect of all, assuming he can afford the hospital, assuming he can afford the antivenom. Is that antivenom actually going to do any good? And that's going to be a lottery. Now, if he's lucky, the hospital will have an antivenom that's raised specifically against the venom of Echis ocellatus, in which case the chances of death are something of the order of 2 or 3% statistically, which in most cases means those patients who get to hospital too late for any help. On the other hand, there's a good chance in many West African countries that he may receive an antivenom that's been raised against this snake here, the Asian saw-scaled viper, Echis carinatus, same genus, both called saw-scaled viper, very similar, used to be considered the same species. Unfortunately, the antivenoms raised against this venom simply don't touch the venom of Echis ocellatus, and the chances of a fatal outcome rise to something like 12 to 15%, which is virtually the same as if you did nothing at all. And why is this happening? It's happening because of variation in venom composition, even between closely related species, which is basically the scourge of snakebite treatment worldwide. It's really why we're having this session here. So I'm sure everybody here knows that venoms, snake venoms, are very complex cocktails involving tens or even hundreds of individual bioactive proteins acting in synergy. And this great complexity generates the raw material for variation in venom composition, and that happens at all levels. It happens in individuals through time, between individuals, between populations, between species, between higher order taxa. It's a ubiquitous phenomenon. So my interest really is in addressing the question, how and why does this happen? Why is there so much variation in snake venom composition? Is there any way that we might be able to predict this? So what determines snake venom composition? We can look on the one hand at the genetic mechanisms that underlie snake venom. So obviously they're proteins, they're encoded by the genome of the animals. So you could have structural changes in the genome, copy numbers, gene presence, absence, and so on. And then the various parts of the chain from genome to proteome are all places where different factors may come in and affect the outcome of that process and the final product of venom. But okay, those are the mechanisms, but what drives these mechanisms? Why do we end up with so many different venom types? And to understand that, the first thing we need to understand is that venom is part of the interface between the snake and its environment. The snakes use venom in their interactions with their environment, specifically with other organisms, primarily prey items or potential predators, adversaries that they need to repel. Okay, so the venom is exposed to the environment around them. It's therefore exposed to natural selection. And that means that natural selection is likely to drive the evolution of venom composition. And if we want to understand how that works, we need to understand the selective factors that drive the evolution of venom composition. So natural selection can be for things like diet, self-defense, the two main uses, but then possibly other factors as well, such as digestion and potentially other factors as well. And I'm going to go through those and look at the evidence that these are important in the evolution of snake venom composition. So if we start off with diet, snakes are predators. They use venom to overpower their prey, to immobilize their prey. That results in multiple different selective pressures, reciprocal. So the snake is under selection from the prey to be able to overcome the prey, and the prey is under selection from the snakes to try to evade the toxic arsenal of the snakes. And we now have quite a bit of evidence that natural selection for diet, for better venom better suited to the preferred diet, is an important part of the selective forces behind snake venom evolution. So we have various patterns and correlations between diet and venom composition. For instance, specialist snakes that feed on a very narrow range of prey tend to have simpler venoms than generalist snakes that feed on a larger number of different prey items. And here's a case study in rattlesnakes by Holding et al., looking at 3 different toxin families. And in each case on the horizontal axis, you've got the phylogenetic breadth of the prey spectrum of different species. So some rattlesnakes eat only mammals, others eat mammals and reptiles, others eat mammal, reptiles, amphibians, and maybe centipedes. So on the horizontal axis, the diversity of the prey, on the vertical axis, the complexity of the toxins. And, in each case, you find that there's a strong positive correlation, except for one of them, there's a strong positive correlation between the complexity of venom composition and the breadth of the prey spectrum, which makes sense. You might need more different toxins to overcome more different prey. Other evidence comes from functional assays, where we've been able to establish that snakes that specialize in a certain diet have venoms that are particularly effective against that diet. One example of that is the saw-scaled vipers, genus Echis. These were studied by Barlow et al. some years ago. Basically, among the different species of Echis, some have diets that consist to a large extent of invertebrates, primarily things like scorpions and centipedes. Others eat primarily vertebrates. And what you can see in this diagram here is the LD50 of different Echis venoms to the scorpion, Scorpiomaurus, and the 2 heavily arthropod-eating species, Echis pyramidum and Echis carinatus, have highly lethal venom to Scorpiomaurus, whereas Echis coloratus, that feeds primarily on vertebrates, actually has a very weakly lethal venom for Scorpiomaurus. And if we map that onto a phylogenetic tree, what we actually find is a very neat pattern where most vipers feed primarily on vertebrates. Echis seem to have switched to an arthropod-rich diet and an arthropod-specific venom early on in their evolution. Most species have retained that, except in Echis coloratus, this was then reversed. The species reverted to a primarily vertebrate-rich diet and lost the specific arthropod lethality that the other species have. So a nice example with 2 instances of coevolutionary change between diet and prey-specific venom lethality. So by now, there's actually a large number of studies which have established these associations between diet and specific lethality or correlation between diet and venom. Quite a few examples of that. We know of plenty of even individually prey-specific toxins. And of course, there's also the flip side of that, which is prey developing resistance to snake venoms. There's plenty of evidence of that, leading to the likelihood of predator-prey arms races where the prey and the predator are playing a game of cat and mouse, trying to evade each other's chemical weapons or each other's defenses. So it seems to be the case that venom variation in many cases is driven by selection for diet for prey. However, I think it's important to realize that that's not necessarily universal. There are other forces at play. And I will just have a brief look at some of those. And one of those, of course, is defense. Now, snakes are part of food webs. They're somewhere in the middle of these food webs. They're predators themselves, but they're also prey for a variety of other animals. And again, we have natural selection at prey. These predators exert natural selection on the snakes to either repel the predators or avoid them altogether. And there's natural selection on the predators to avoid the chemical weapons of the snakes through either resistance or through behavioral means. Now, in terms of the importance of defense as a driver of venom evolution, we have an awful lot less data than on diet. What do we know about the use of snake venoms in defense? An awful lot of that is from snake-human conflict. Snake bites to humans are usually the result of the snakes using venom in defense, and that's what we've studied. Studying the role of defense in the evolution of venoms has generally tended to be more difficult. The predators of snakes tend to be quite large vertebrate predators. It's very difficult logistically and ethically to do a lot of experiments on there. So until, by and large, still the importance of defense in driving the evolution of venom isn't particularly well understood. How can we test the defensive function of venom? Well, the obvious one is to think, okay, what would you expect a defensive venom to do? How do you use a venom defensively? And if we look at other animals which have venoms that are primarily defensive, the main thing they do is they cause pain. How many of you have been stung by bees and wasps? A fair few, most of you, I would imagine. What happens when you get stung by a bee and a wasp? It hurts. It hurts straight away. You don't get stung by a wasp, and half an hour later you think, oh, this is starting to hurt a bit. It hurts straight away, because that's what an animal defending itself wants. It wants to repel you before you've caused it too much damage. And if you want to see some really nice displays of what defensive venoms can do, go to YouTube and do some sort of search for scuba divers stung by lionfish and things like that. There's some very interesting videos of scuba divers screaming at the top of their lungs all the way to the surface through decompression stops and all the rest. So that's what you'd expect a defensive venom to do. What about snakes? Do we see much of that? Now, reading through the literature in general, it's a very, very mixed bag. On the one hand, we have species such as the genus Bungarus, the kraits, found in Asia, and these are actually notorious for causing painless bites. People get bitten in their sleep, they either don't wake up, or they wake up, but it doesn't hurt, there's nothing to see, so they think, okay, nothing happened, and they go back to sleep, and then they wake up close to death. But then on the other hand, we have other species like the Texas coral snake, Mycorrhiza tenaire, which actually has specific pain-inducing toxins. And by all accounts, these bites are very severely and rapidly very painful indeed. That's a bit of a mixed bag. One of my students, Harry Ward-Smith, tried to sort of establish whether there was a more general pattern with a survey of our fellow herpetologists who do get bitten by an entertaining variety of snakes as part of their job, and basically simply did a survey of the pain trajectories experienced by these snakebite patients. And the basic prediction is that if a venom evolved primarily for the purpose of foraging rather than defense, you would particularly expect severe pain as an early symptom. The snake doesn't care if the prey falls asleep peacefully or if it dies in agony, it just wants to eat it, okay? On the other hand, if a venom evolved largely or fully for the purpose of defense, then you would expect this very rapid early increase in pain, where the peak pain would be very early on, would be reached very early on in the course of the envenomation, within the first few minutes, before the snake is so damaged that it doesn't help the snake anymore if its predator is in pain. If you look at the results across the various families of snakes averaged out, what we're seeing by and large is more the first pattern, which is that the maximum level of pain was reached long after those ecologically relevant initial minutes where it would benefit the snake to inflict severe pain. And that's across all the 4 families of snakes included in the study. And that applies even to species of snake which are notorious for causing really unpleasant, painful bites. So this is Bitis areatans, the African puffider on the left, notorious for horrendous bites with major tissue damage. And on the right, the Western diamondback, Crodalus atrox, again, really notorious for causing horrendous bites. And yet, when you look at these graphs here, the individual colored lines are the pain trajectories of individual patients. The dashed lines are the average. And what you can see in both of these species, during the first 5 minutes when it would be relevant to the snake for self-defense, the pain wasn't all that bad. It got really bad later on, but it wasn't particularly bad in the first few minutes where you would expect it to be bad in a defensive venom. And here's our own European adder, Vipera berus. And you can see the same general pattern from the dashed line. And the other thing you can see is just how variable it actually is. There are some of these envenomed bites which cause no pain whatsoever from the beginning to the end, whereas others were obviously very severe right from the start, and others were very mild early on and became very severely painful later on. A huge amount of variation. That's not what you would guess, but that's not what you would get if you did the survey about wasp stings or lionfish stings or things like that. Okay, so, and finally, what was really remarkable is when we asked how long did it take for the bite to become so painful that it was incapacitating, only 15% or so said within the first 5 minutes, which is where you would expect it to be for defensive venoms. And in fact, more than half never actually got that painful. So if you put all that together, that kind of suggests that there certainly isn't a broad general trend of venom evolution being driven by the need for defense, because then we would see far stronger evidence of common early pain. Are there exceptions to that? Well, the first place to look at would be at snakes that have specifically defensive adaptations relating to venom. And the obvious one for that is in spitting cobras. Spitting cobras can project venom for a couple of meters or so out of their mouth by compressing their venom gland. And if it gets into the eye, it is known to be atrociously painful. It's a purely defensive adaptation. They bite their prey just like any other snake. And interestingly, this mechanism evolved 3 times independently in African and Asian spitting cobras and in the ring-cowled Semicartus in Africa. So it's a nice 3 times replicate experiment. And we know of the behavioral and morphological adaptations. What about the venom themselves? A couple of years ago, with colleagues from Liverpool School of Tropical Medicine and a wide variety of additional collaborators, we looked at that. In this diagram, we've simply mapped the extent of nociceptor activation onto the phylogeny of cobras. Basically, the warmer reddish color, the more warmer and reddish the colors, the more painful, the more effective these venoms are at triggering nociceptors. And what we can see is that the most painful venoms are associated with the three spitting clades. Hemacartus at the top, is it showing up? Okay, Hemacartus at the top, the African spitting cobras here, and the Asian cobras down there. If one looks at the venom composition of cobras and the different groups of cobras, the main thing that stands out is that the 3 spitting lineages have venoms that are particularly rich in phospholipases A2, PLA2s. And that leads to the question, okay, what role do the PLA2s play in causing the excess nociceptor activation in spitting cobras? And if one starts to separate out the different venom components and recombine them to find out what's causing the pain, what you find is that the PLA2s actually don't do anything. Okay, they don't cause nociceptor activation. The usual suspects for causing pain are cytotoxins that disrupt and are toxic to cells, which you might expect. It turns out if you look at the cytotoxins on their own, they do activate nociceptors, i.e. cause pain, but not to the same extent as the entire venom. But it's when you combine together the PLA2s and the cytotoxins that you start to recapitulate the full effect of the venom. So it's a synergy between these two toxins that causes the increases in pain in spitting cobra venoms, which is of course advantageous if you're relying on your venom for defense and you want to quickly repel your adversary. So we've got synergy between different toxin types, and interestingly, it's the same across the three lineages of spitting cobras. The same mechanism has evolved 3 times independently. So here we've got a clear example of natural selection for defense associated with a particular defensive adaptation actually driving the evolution of that part of venom composition in these cobras. But is there more there than that? Here's another example of a snake with really quite extreme venom variation. It's the Mojave rattlesnake, Crotalus scutulatus, from the southwestern US and Mexico. These snakes have 2 radically different venom types, termed venom A and venom B. Venom A is highly neurotoxic, has very little in the way of tissue damaging or hemotoxic activity. Venom B, on the other hand, is more your classic rattlesnake venom, rich in metalloproteinases, causing blistering, tissue damage, and all the rest. And these venoms differ grossly in their mode of action, neurotoxin versus not. And in their lethality, in mouse assays, venom A is 10 times as lethal as venom B. And you can find these 2 venom types literally by driving half an hour down the interstate. And if one starts to look at the diet of these different populations, in this diagram here, the inner pie chart is the diet. And it turns out most of these populations are very similar. They all eat round about 2/3, 3/4 mammals, and 1/4 or even less in the way of reptiles, quite independent of what venom type they have. So no real evidence of relationship with diet. And if one starts to run some statistics, in this case mental tests, what you get, this is a heat map of what factors seem to be associated with venom types. It turns out that it's got nothing to do with obvious null models like isolation by distance or the genetic distance between these populations. Notably, it's got absolutely nothing to do with diet. It's completely independent of diet, irrespective of whether you look at diet at the level of family, genus, species, overlap, or class. It's got nothing to do with diet. Looking at ecological factors, it's got nothing to do with topography, with vegetation, with ecoregion. The one thing it strongly correlates with, interestingly enough, is actually climate. And in particular, of the bioclim variables used for niche modeling, the ones that came out as most significant were annual mean temperature and mean temperature of the coldest quarter. So these are the main factors that correlate with venom composition in the snake. How does climate determine venom composition? Does it? I don't know. I'm not sure I believe it myself. I suspect there's some other prey-related thing that we're overlooking that's correlated with climate in itself, but that's what we're getting like that. Okay. Moreover, it turns out that in this species, rather than diet complexity and venom complexity being correlated, it's actually the other way around. We find that Mojave populations with broader diets actually turn out to have less complex venoms. So these snakes are doing something completely different than what we would predict. So there are plenty of mysteries still to be solved there, how exactly this works. So in terms of selective drivers, we have a very wide range of selective drivers, including diet in various different ways, defense, and possibly even things like climate or other things associated with climate. And it's perhaps unsurprising that these very complex and often interacting selective drivers really result in complex patterns of venom variation. We can then also look at the issue of the genetic mechanisms that underlie venom composition, sources of variation. As in any proteinaceous secretion, we start out at the genome. Sources of variation are things like presence, absence of entire genes and loci, copy numbers of genes, point mutations, and of course the entire system that controls venom secretion. From the genome, we move on to the transcriptome. And here we have a possibility of selective transcription of existing toxin genes. Just because it's in your genome doesn't mean it gets transcribed into messenger RNA. And from the transcriptome, we move on to the venom proteome, and here there's scope for additional variation to be produced by selective translation of messenger RNAs. And finally, from the venom proteome to the final phenotype, we can then still have post-translational modifications of proteins that have already been secreted before we come to the phenotype, which is of course what comes out of the fang and is then exposed to natural selection. If we now look across the diversity of snakes, it turns out that all of these various sources of variation apply in various cases. So this is a diagram showing the evolution of metalloproteinases in rattlesnakes. At the top of the slide, we have the king cobra with the ADAM28 genes and then the NEFM and NEFL genes. And in between, the king cobra has managed to produce 2 genes coding for metalloproteinases. Meanwhile, here's the western diamondback that's got those genes either side, ADAM28 and NEFM and NEFL, and it's got something like 40 different genes for metalloproteinases produced here. So this is all genome level changes resulting in differences in venom composition. And that applies even among rattlesnakes. For comparison, here's the western diamondback, Crotalus atrox. Here's Crotalus scutulatus venom B, the one that causes tissue damage and all that. And you can see that a whole bunch of metalloproteinases that are present in Crotalus atrox aren't present in scutulatus venom B. And then in venom A, the neurotoxic version, most of them, in fact, are absent. There are very few metalloproteinases left in venom A, Crotalus scutulatus. So even within that species, we have major differences at genome level in terms of what genes are present and which ones aren't. And in terms of how those genome level differences translate into differences in the phenotype, in what comes out of the fang, it turns out that in Crotalus scutulatus, that's actually a very direct link between genome and phenotype. So this was an early attempt to demonstrate that. We're using Mojave rattlesnakes with and without Mojave toxin. So of the 2 here, the ones on the outside are Mojave toxin positive. The ones in the middle didn't have it in their venom. That's a PCR gel. If you do a PCR for that gene in Mojave toxin positive snakes, you'll get a PCR band, a PCR product. If you do it in Mojave toxin negative snakes, you don't, because they don't have the gene. It's that simple. And moreover, when you test for the correlation between proteome and genome, you find there's an absolute association. So here in this diagram, each of those vertical series of bars is one specimen. The bottom two rows here are what's in the genome. So black means that the gene was there, gray means it wasn't. On top here, that's the data from the proteome where we had it. And what you can see is basically whenever we had a proteome for a snake with Mojave toxin in the genome, it also had Mojave toxin in the proteome. Very simple. If it's in the genome, it's in the proteome. And it turns out that the same also applied for all the other major venom groups in the Mojave rattlesnake. Very simple. It's in the genome, it's in the proteome. You just need to do a PCR on a roadkill and you already know what venom it had, even though its head was chopped off. On the other hand, here's another example. This is from a paper by Kartik Sunaga et al. From a couple of years ago. This is looking at different populations or taxa of the crates, Indian crates, Bungarus ceruleus complex. And it turns out that the 3 different populations he looked at actually had very similar transcriptomes, okay, dominated by PLA2s and 3-finger toxins, as you would expect in elapids. However, when you then start to look at what comes out of the venom gland, which of these messenger RNAs have been translated into proteins, we get staggering differences in the abundance of different toxin classes, with one population being heavy on 3-finger toxins, another on PLA2s, and another also on acetylcholinesterases, okay? So really similar transcriptomes. They obviously have the same genes, and they transcribe the same genes, and yet when it comes to actual translation and expression in the proteome, they're completely different, to the point where they're very different lethality, and antivenom cross-neutralization is a major problem. And perhaps the scary part of this is, if all these crates have all the genes, here we've tried out 3 different populations, but really if they all have the same genes, the scary bit to me is you could get these kind of differences on a much smaller geographic scale, because really it's just a matter of gene regulation, which genes are expressed. So I think there's a lot of research needed to what extent that is a problem among crates in their range. And then finally, somebody else does something different. This is the puff adder, and this is basically the genes through to finished proteome chain in the puff adder, published by Casewell et al. 2014. And you have partial transcription of toxin genes. They're genes there that are never actually transcribed. Then you have selective translation. A whole series of messenger RNAs are never actually translated into proteins. And then you have the additional layer of post-translational changes, where even the proteins that have originally been secreted acquire an additional level of diversity through post-translational changes, which adds considerable additional complexity to the venom. So the puff adder doesn't just do one of these different things, it does all of them. So when we look at the genetic mechanisms as sources of variation, we have great diversity. Rattlesnakes, if it's in the genome, it's in the venom, pretty simple. It's just what's in the genome that differs. Crates, they got very similar genomes, but produce completely different proteomes thanks to selective translation. And then we've got the puff adder, which does a bit of everything. So not only do we have complex interacting sources of selection that act on venom composition, we also have a whole series of different mechanisms which control what actually comes out of the fang at the end. Now, of course, the thing to remember is natural selection for venom composition acts at the level of the phenotype. Natural selection acts when fang hits flesh, when toxin hits receptor, okay? And this is what then feeds back to all these genetic mechanisms that determine venom composition. So at one level, if we want to understand how the system works, we really need to understand the selective forces. And really, although we've got some examples now of diet or defense being important, we really need to know a lot more about the natural history, the ecology, behavior, the interactions between snakes and prey and predator, and the venom effects of on prey and predator in order to really understand what drives this in different examples. And there we have absolutely huge knowledge gaps. And we also have a huge kudos gap. It's an awful lot easier to get venom to do proteomics or genomics or whatever than it is to go out and study the natural history of a snake. And that's a major knowledge gap. And I think it's also one case where citizen science, with at least observations of interaction in nature, can really fill a lot of these knowledge gaps, at least to some extent. So why is it also complicated? Well, because it is. We've got multiple sources of selection acting on venom, in turn acting on multiple different genetic mechanisms, which affect the final product of venom composition. It is complicated. There's no way around it. And for those of us who are interested in antivenoms and things like that, the bad news from that is that really there's no easy way of predicting what antivenom is going to work for what, which particular snakes are going to have very different venoms and which are going to have very similar venoms. There's no shortcut to that. We really do need to look at an awful lot of venoms to understand what's going on in these medically important species, which are causing so much death and disability worldwide. We need to be aware of oversimplifications, okay? One thing that always amuses me in the proteomics lecture is that you get papers where they produce one proteome and do some antivenomics on top of that from one specimen of a species. And then the next bit sort of says, we have done the proteome of species X. Sorry, but you haven't. You've done the proteome of one individual of species X. At the moment, we have no real idea to what extent that individual represents the total envelope of venom diversity, both ontogenetic, individual, or geographic in that species. So we need to be a bit careful about these seemingly simple oversimplifications. So really, in terms of antivenom development, I think, yeah, once upon a time I would have stood here and told you how important taxonomy is. Really, what really needs to happen is we need to identify the major venom phenotypes, which may correlate with species or phylogeny or not, and identify the major important venom phenotypes that we need to address with antivenoms, and we need to just do that experimentally. We can't assume that we know anything from other factors. And okay, I'm not a physician, I'm not a pharmacologist either, but if I were to put money on where major breakthroughs in snakebite treatment are going to come from, they're probably going to come from novel approaches that are more broad spectrum, that are less affected by this very fine-grained variability in venom composition. So repurposed small molecule inhibitors undergoing medical trials. Certainly the lab trials that have been done suggest that they're really promising in neutralizing the activities of entire toxin families across a broad swathe of venomous snakes. And similarly, sort of looking at producing synthetic recombinant antibodies against the generalized features of entire toxin families is likely in the longer run to probably result in more breakthroughs than working with the conventional antivenoms that we have today, which of course remain essential in the short and medium term and will probably also remain important into the future. So with that, I have an awful lot of people to thank who I've worked with over the years and who've sort of shaped my thoughts on the topics that I've been talking about today. The Leverhulme Trust for indulging my fascination with snake venoms and venomous snakes. And finally, you all for your attention this afternoon. Thank you very much.